I've received a lot of questions about the idea of an individual with high glucose ending up with glucose in their urine, and so we're going to deal with that portion of that learning objective in this video. So why is glucose present in the urine of individuals with hyperglycemia? First, we just need to figure out what hyperglycemia means. So hyper is just too much, so high levels. Glycemia is the concentration of glucose in the blood. So why does somebody with high levels of glucose in their blood end up with glucose in their urine? Normally, urine does not contain any glucose. So remember, when you get confused about something in the kidney, think about the big picture. Kidneys are in the body to filter the blood and remove waste products and create urine. And this is happening over in the renal corpuscle. This is where filtration occurs. But filtration in the kidneys is based only on size. So anything small, like waste products, will end up moving from the blood into the renal tubule. But other small things, such as glucose, are also going to make this trip. The amount of any solute that shows up in the renal tubule is going to correspond with the amount of that solute that was in the blood. So if there's lots of glucose in the blood, lots of glucose will end up in the filtrate. So how do we get back the glucose that we have filtered? Because it's not going to end up in the blood normally. So here we are in the filtrate where we have some glucose. We also have waste molecules and things like sodium and water. And on the apical membrane of the cells of the proximal convoluted tubule, so this is a PCT cell, we have these transport molecules. And the ones that we care about in this video are glucose-sodium symporters. That means glucose and sodium will travel together through these molecules into the cells of the proximal convoluted tubule. Once this happens, glucose can then travel through the basal membrane using facilitated diffusion. So there's a transport molecule that will just allow glucose to go from the cell, out of the cell, and then it will make its way into the bloodstream again. These transport proteins, the symporters, are kind of like a revolving door for two reasons. One reason is we have a limited number of doors on any building. This is the same as the limited number of transport proteins on the proximal convoluted tubule. The other thing that's the same is for each one of these doors, there's a limited number of people that can go through in any amount of time. So we're going to say there's a small number of solutes, small number of people, which is the same thing as solutes, can travel through at once. So using people in this door, let's talk about what this would look like. So if there's a stream of people walking by this door, and there's just one individual at a time, this person can easily travel through the door and make it outside. You'll have to use your imagination to see the door spinning. It's not a GIF. But if we had more than one person at a time and they came upon this door, not all of them can fit into that little spot at once. We can only move one person through. The other people, they just have to keep walking. And so they will be stuck inside that building like molecules in the nephron would be stuck inside the renal tubule. Okay, so now we're back to the kidney cells, having learned a little bit more about how these molecules, these transport proteins, are working like a door. So if there's a whole bunch of glucose inside of the tubule, but only one can go through at a time, we'll see that one glucose leave, but the other ones will have to continue through the tube. As we travel further and further down the tube, we're not going to have any more of these transport proteins. Almost all of our reabsorption of nutrients is happening up here at the proximal convoluted tubule. So if the glucose doesn't travel out of the tube when it's in the PCT, it's just going to continue through the rest of the tube and end up going out here to the ureters, to the bladder, to the urethra, and being eliminated. So that's kind of the logistics of everything that's going on. Now let's use the actual definitions for these terms. So the renal threshold, this is the plasma concentration, so the concentration of a solute in the blood at which a solute will begin to appear in the urine. Remember, the amount of any solute that shows up in the renal tubule is going to correspond to the amount of solute in the blood. So if we have lots of glucose in the blood, we'll see lots of glucose in the tubule. And if that's above the renal threshold, there will be too much for us to reabsorb. There'll be too much to go through that revolving door. The other term that we introduced was the transport maximum or the tubular maximum. Your textbook uses the tubular maximum, but transport maximum is the same thing. And this is looking at a rate. So the rate at which a substance can be actively reabsorbed. So this is a quantity per minute. So imagine that each one transport protein can move one glucose every 5 seconds, right? So we could move 20 glucoses per minute. If we exceed either the renal threshold or the tubular maximum, whether we're talking about concentration or rates, these solutes that normally would be completely reabsorbed are going to start showing up in the urine. I hope the door helps make that a little bit more clear for you.